Right. Hello, everyone. So I'm actually on vacation right now. It's summer, and well, I'm bored, and so I decided to make this lecture, and I've been wanting to make this lecture for a while. So yeah, I'm pretty excited about it. And okay, so we're going to be talking about gene regulation today. So the control of which genes in our genome in the cell, the collection of our DNA, the entirety of it, which ones are expressed at certain points in time and which are not expressed, which are turned on, which are turned off, and how the cell does this in response to internal signals, external signals, and why it would do this. And part of why the cell does this, well, think about, or maybe you have thought about how skin cells are like these flat barrier type cells that serve a protective role, and they're packed with the protein keratin. It provides the structural support of the skin. And maybe you've thought about, neurons, which are the cells of the brain and the nervous system. They look really kind of like spiders almost. They're really interesting looking. And of course, they're making different types of proteins than a skin cell would, involved in, how I move my hands and how I feel pain, or, all of these types of signaling involved in the nervous system. So what about a pancreatic cell? Pancreatic cells also look like different. They're cuboidal, we call them. They're glandular cells. They are producing digestive enzymes. So point being is all of these cells look very different, and they act very different. And so we're going to look at the mechanism. How does this happen? And today we're going to focus on, excuse me, we're going to focus on the gene regulation, the regulation of gene expression in prokaryotes. Okay. Excuse me. So recall that our prokaryotic organisms, these are our single-celled, right? Single-celled microbes, are microorganisms that lack a true nucleus. They don't have membrane-bound organelles. Very simple, very, very small, right? Like 1/10 the size of a typical eukaryotic cell. So these are our bacteria. Bacteria are prokaryotic, unicellular, right? They're no multicellular bacterial organism. They're just independent, single-celled, right? Our other branch of prokaryotes were the archaea. Again, another single-celled prokaryote. So today's lecture, we're focusing on the regulation of gene expression in prokaryotic cells. And then next time we'll look at gene regulation in eukaryotes, like ourselves. Ourselves. Okay. Okay. without further ado, let's get started. All right. All right. So I don't have my whiteboard, but we will prevail, I assure you. Okay. So let's begin. So before we dive into prokaryotes, I like to sort of mention the importance of gene regulation. There's a lot actually, but really the most striking and obvious would be, again, how these cells appear and how they function very differently, right? This all has to do with which genes are being expressed, which proteins are being produced, because the proteins that a neuron would produce, it's very different than what skin cells are making. These skin cells here, this is a histological stain cross-section of the skin, and all those skin cells, you can see all the nuclei. They're flat. We call them squamous cells. They're flat, they're packed together, they form a really incredible barrier to conserve and protect everything inside, prevent water loss, etc. But they look very different than our neuron here. Here's our neuron that it's like I said, a spider with a tail. I don't know if I like that example, but it's maybe like roots of a tree, actually. You know, it's very interesting, right? And so neurons are producing these neurotransmitters that travel down this axon to communicate with other neurons and eventually send a signal, like if I want to move my arm, or if there's I touch a hot stove, that pain message is relayed to my brain through these neuronal connections. But again, they look very different, act very different. So structure and function, right? Very, very different. And then here are some white blood cells. The eosinophil doesn't look white, but it's because of the specific stain. But these are, again, look very different, and they're producing all types of different proteins involved in the immune system. The white blood cells function in the immune system to defend us against pathogens, against viruses, fungi, bacteria. And so if you take micro and we talk about immunology, you kind of learn that all of these different white blood cells have specific mechanisms to essentially destroy, eliminate these foreign invaders. And this all has to do with gene regulation, which genes are being expressed in these different types of cells and which are not. You're not going to see a neuron producing keratin that a skin cell would produce. It has no use for our neurons, right? And their related function. You're not going to see our skin cells making enzymes involved in destroying bacteria. That's the white blood cell's job. So that's kind of the overall theme here. Okay. So importantly, we all start as one cell. And through successive mitotic divisions over and over and over, of course, we form us. But we learned in mitosis that we are replicating our DNA to produce identical cells, identical daughter cells. And so all of our cells contain the same DNA. They have identical genomes. So again, how can they look and act so differently? Well, importantly, skin cells, white blood cells, they have the information to become a neuron, to act as a neuron, produce proteins that a neuron would, but they don't, right? They silence those genes, is what we call it. They're turned off. And the proteins that is related to their function are turned on. And so we're going to learn how this happens. And through development, actually, from that single cell, gene regulation comes into play as organs form. Because once they're formed, the structures and functions are becoming defined in the organism. But again, today we're focusing on prokaryotes. And of course, different cell types doesn't really apply to a single-celled organism, right? But nevertheless, there are mechanisms of gene regulation that are very important in bacterial cells and archaea. And so we're going to talk about them first. And they're different from eukaryotes. And eukaryotes we focus on next time. Okay. So, all right. So again, no whiteboard, but I'll just read out loud what I'm writing so you can write it in your notes. First thing I want you to write is all organisms, both prokaryotic and eukaryotic, regulate gene expression in response to internal and external signals. All organisms regulate gene expression in response to internal and external signals. Okay. So let's define differential gene expression in words here. Let's define differential gene expression. It's kind of what we were just discussing. That different genes are being expressed in a brain cell than a liver cell, than a kidney cell, muscle cell, etc. So differential gene expression refers to, let's write, in multicellular organisms with specialized cell types. There are some organisms that are multicellular, but they're just a big blob of the same cells, certain protists and stuff, besides the point. Most have specialized cell types, plants, animals, etc. So specialized cell types, blah, 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 where are we going? Each cell type maintains a specific program of gene expression. So again, all the cells in my body, the somatic cells, have identical genetic sequences in the nucleus of each cell. So why aren't they all the same? This is because of differential gene expression. My neurons contain the information to produce digestive enzymes, as a pancreatic cell would, but it doesn't turn them on. It only turns on the neuron-specific genes. So we're going to learn about the mechanisms of how this happens. How do we turn genes on and produce the final protein product, if it's an enzyme or a structural protein like keratin? And we're going to learn how they suppress, silence genes, turn them off, keep them off. They're not supposed to be making them. And we're also going to talk about why is this useful? Why wouldn't we want to be making things all the time? It has to do with evolution. So we'll talk about that. It's pretty interesting. Okay. So again, let's look at a few classic examples of gene expression in prokaryotes. There's these things called operons, again, specific to prokaryotes. And the first operon is the Tripoperon. Tripoperon. Trip is the gene that encodes for the amino acid, or actually, I'm sorry, it encodes for the enzymes involved in synthesizing the amino acid tryptophan. So we're going to talk about this. Okay. The Tripoperon, E. coli, bacterial cells. So it's a very highly studied model organism for molecular biologists. They use E. coli a lot. And so E. coli contains the Tripoperon. And so trip is the gene. So we italicize it, it's lowercase. So the first thing we're going to write here about the Tripoperon. We're synthesizing tryptophan. We're creating it, building it. So it's a biosynthesis pathway. So our cells, some cells are actually able to make amino acids from other precursor molecules. So this is just a classical example of gene regulation. So we'll see. It's a pressable, right? Yeah. An example of a repressible, something that can be turned off. So we'll talk about that. Tryptophan, this is what we're going to write. Tryptophan synthesis pathway. And so this will be in our, sorry, not our, right? This is prokaryotic pathway. E. coli. I say our because, funny enough, if you've ever heard of the gut microbiome, it's just massive emerging field of research. We have trillions, 10 billion, a lot of bacteria that live in our large intestine. And they have a lot of important roles, beneficial roles, actually, to nutrient absorption, to vitamin synthesis, defending against other bad bacteria, things like that. But E. coli, there's actually good E. coli bacteria. It's not the pathogenic strain that makes us sick. And these E. coli bacteria, so E. coli bacteria, gotta be picky. I'm a biologist, so I want to write the organism correctly. So I italicize, and it's a capital E with the lowercase coli for the genus and the species. E. coli bacteria that reside in our colon synthesize the amino acid tryptophan when our diet doesn't supply it through, kind of a long sentence, but whatever. So tryptophan is not supplied through our diet. So it doesn't supply it through a biochemical pathway whereby each reaction in the pathway, and we've seen examples of these types of biochemical pathways, right, in metabolism. Same sort of idea. This would be more of an anabolic process. We're building a molecule, right? Adding chemical groups in a series of reactions, each catalyzed, so by an enzyme, right? A different enzyme. So supplied through a biochemical pathway whereby each reaction in the pathway is catalyzed by a specific enzyme. Okay. Awesome. So again, we defined differential gene expression, or I'm sorry, we're going to regulate our genes in response to internal and external signals. So it kind of links signal transduction, which is another sort of topic in biology, but the external signal in this case is that our diet, we're not eating enough protein. The tryptophan amino acid isn't being supplied. So what's the E. coli going to do? It's going to produce tryptophan. Let's do a quick little reminder here. Oh, actually not yet. There's one more note. And so you guys will actually have, you'll have all of these slides. I confess that it's a little more PowerPointy than I would have hoped when I made it, but I really wanted to give you the whole picture because I think it's going to explain it better. I think you'll have less confusions, in my opinion, because this is kind of confusing stuff. It's a little abstract, but so essentially, we're going to be producing tryptophan in the absence of it when in our diet. Okay, not we, the E. coli, right? Now, bacteria, they have one chromosome, right? They don't have much genetic material. They're so small, they don't need 20,000 genes like we have. And so on this chromosome, the 5 genes, there are 5 genes that code for these enzymes in this pathway. And they're actually one after the other. They're clustered together. So when this DNA is transcribed into mRNA, it's one long mRNA molecule that codes for 5 separate proteins, enzymes, polypeptides, right? Same thing. It's a fancy word for proteins. And I just included the note. Now, how does this happen? I thought it was like one gene, one mRNA, one protein. Well, essentially these 5 genes that are all transcribed together, they each have start and stop codons. So when that mRNA reaches the ribosome and is being translated into these protein products, these enzymes, they have separate start and stop codons, which actually signal the ribosome to begin a new protein and this old protein for all 5. So it's kind of funny that the prokaryotes are a little more efficient in this way. They're a little, kind of the cheap, easy, fast way to make proteins. Like, let's do it all at once, right? Not have separate mRNA strands, separate genes. That's more of what we do. Okay, so here's a really cool picture of this pathway. And so if we start on the left side, this is sort of the schematic of what a biochemical pathway looks like. As a reminder, it's sort of these molecules, precursors that are modified through a series of chemical reactions, each of which catalyzed by a different enzyme to produce that final product of tryptophan in this case. Okay. Now, as you see, these are the genes, TRPE, DCBA, all these separate genes that are right next to each other in this Tripoperon, we call it. They code for these enzymes. If you see 2 genes, then the enzyme has multiple components to it that come together to form the entire enzymatic complex. The entire enzyme requires multiple polypeptides. Don't worry too much about that, but just that these genes are responsible for the production of the enzymes, and therefore the tryptophan can be produced when we activate the gene expression here. And we'll talk about this negative feedback in a moment. Okay. Just want to stop the recording for a minute and see how long it's been. Okay, we're all good. All righty, so before we look at the Operon with these genes, more vocabulary, more terms. Again, we use a lot of words in biology that are, not typically used in everyday life. So these words, we have promoter sequence, promoter sequence, so the promoter is the site where RNA polymerase can bind to DNA and initiate transcription. Remembering that in transcription, we are transcribing DNA to produce an M messenger RNA complementary strand. So that's the promoter. Promoter is where RNA polymerase comes in and attaches to DNA and will activate transcription. The operator sequence pen is dying. Hey, maybe it's a sign I can just use a different color. Let's do it. Do that in use blue operator sequence. So the operator, this will make more sense when we look at the operon. This is the on-off switch that controls access of RNA polymerase to the genes to be transcribed. So our operator is really the control switch. It'll either allow RNA polymerase to transcribe these genes or prevent RNA polymerase. And lastly, green. So together, the operator, the promoter, and the genes they regulate, this is the operon as a whole. The genes they regulate constitute an operon. Okay. Wonderful. Let's take a look at this operon. Actually, before we do, I'm going to explain how it works. Let me see. Oh yeah, I'm going to draw it. Huh, cool. I was going to include a picture, but actually I can pull up the picture after we draw it, if it's going to, be helpful. Let's see here. All right, let's draw it first. Kind of working on the fly here. I think that's a better idea. We're going to draw this operon first. Okay. Oh, yeah, blank. Ha! All right. So let's do this. Oh, I'm going to need my, hold on, let me pause. I'm going to get a pen that works. All right, this one should be good. All right. So first thing I'm going to do is I'm going to draw a little bit of DNA here. Actually, let me this way horizontally. Okay. Oh, that's the other side. All right, now I'm being picky. Let me fix it. Do that again. Okay, so drawing a little piece of DNA. And so now I'm going to begin the start of the operon. I'm going to use green because I think that's what I used to define operon on the previous page. And so again, this is the Tripoperon. So I'm going to leave a little space and I'm just going to draw a green line like that. So I left a little space, then I have a green line. So that green line is going to be divided into 1, 2, 3, 4, 5 genes. Okay, so I did 4 hash marks. I know that's kind of hard to see. So there's 5 segments of DNA encoding the genes of the operon. So this was trip E, D, C, B, A. So let me write that in and then I'll show you. Trip D, trip C, trip B, and trip A. Okay. So again, these are the genes of our operon. That are encoding these enzymes, right, in the pathway to produce tryptophan. Now let's fill in the blank space, okay? The first is the promoter, which we used black, I think. So the promoter, this is where RNA polymerase binds. And often it includes the operator. So I'm gonna but let's draw them separately. So this is gonna be the promoter, and we'll leave that space for the operator, which I think I used as blue, right? So we've got our promoter, operator, genes, okay? The entire thing constitutes the operon. So this is our operon. I'll go back and label these as well. Promoter, and then in blue would be the operator. Sequence of DNA, so it looks like that. Okay, so RNA polymerase, I'm going to do in red, the enzyme. And so let's again, where does that attach? The promoter, right? So as RNA polymerase says, the textbook, it's kind of like a blob sort of thing. It looks like a guitar pick blob. Let me show you what I mean by that. I think it shows it has directionality. There's my RNA polymerase binding at the promoter. And it's, of course, going to travel this way and transcribe our genes when the operon is active to produce an mRNA molecule. So I'll draw that as well. So transcription into mRNA, single-stranded mRNA molecule. And again, there's these start and stop codons between each of the 5 genes. So I'll kind of represent that with the hash marks. And so these mRNA sequences are going to be translated eventually into 5 separate polypeptide subunits that make up enzymes for tryptophan synthesis. Just because I'm picky and I like to do this. Remember, we synthesize mRNA 5' to 3'. So I just added that in as well. Read 3 to 5, build 5 to 3, synthesize 5 to 3. Read the DNA, that is 3 to 5. Okay. And just to complete the picture, for translation, ran out of colors, but I guess we can use green. Actually, let's do black. Black, green, black. So during translation, right? So that was transcription to produce mRNA, but during translation, we're going to be making, just going to do little protein blobs here, making proteins from the mRNA. And remember, translation occurs at the ribosome. And I'm going to even include that these are the protein subunits that make up enzymes for tryptophan synthesis. All right, so far we haven't discussed the regulation of this operon. How is it regulated? So we're going to add that to our picture after we discuss it. I know I might be moving a little fast. It's hard to tell the time. It doesn't really tell me on Zoom where my time's at, but maybe I'll just edit it later. So I can, but it is recorded. So if you missed something to write down, but alas, we can push on. Okay, so how does the regulation come into play? So the Tripoperon can be turned off, meaning we're not going to be producing these enzymes. This repressor protein is what's responsible for that. So when the trip repressor binds to the operator, remember the operator is the on-off switch. The repressor can bind to the operator. When it does that, it gets in the way of RNA polymerase and prevents it from transcribing the genes downstream. So downstream is one of these words, upstream, downstream. We use in biology to refer to something that happens later in the pathway or prior. Upstream is prior, eventually downstream is later. So the Tripoperon we say is repressible because, hold on, where is it? Okay, never mind. We say it's repressible because by itself, the Tripoperon is active. So what we just drew, without anything regulating the Tripoperon or affecting it, it's going to be transcribing these genes. There's nothing in the way of RNA polymerase binding to the promoter and transcribing these genes. So again, by itself, it's active. So we say it's repressible because we can turn off its activity. It's always active, but we can turn it off. And we'll talk about why we want to do that shortly. Why wouldn't we always want to be producing tryptophan? But let's look at the how here, the mechanism. Where does the repressor come from? This repressor that binds to the operator. There's another gene. It's a regulatory gene. It's actually not even close to the operator. It's located it can be on another, I almost said another chromosome, but we're in prokaryotes, right? No, one chromosome, one DNA molecule. But it's further away. It could be hundreds of thousands. They have a tiny genome. So it could be like, hundreds of nucleotides away, this regulatory gene. And so this gene is called TRPR for regulatory. And again, it encodes for this repressor protein, and it has its own promoter. Okay. Now here's where things get trippy. Oh boy, you're gonna have to use that one in the future. The repressor protein is regulated itself. And so the repressor protein is produced, right, from this gene. And hold on, I'm just writing. Okay. The repressor protein can itself be repressed, meaning it won't bind to the operator. We're going to draw all this out. But the thing that represses the repressor, the thing that prevents it from binding, is tryptophan itself. Oftentimes in these biosynthesis pathways where you're generating a final end molecule, it can negatively feed back and prevent more synthesis. Again, we're going to talk about why we would want to do that. But if tryptophan's around, as in from the diet, and the E. coli, there's a bunch of tryptophan around, it's going to negatively feed back, bind to the repressor, and induce this conformational change, meaning it's going to change its shape. And what it does when it does that is it actually has a higher affinity, a higher attraction, a likelihood to bind and attach to the operator and prevent transcription of these enzymes. Okay. But without tryptophan bound to the repressor protein has a low affinity and it won't attach to the operator and therefore will make tryptophan. So you see what's going on here. When tryptophan is around, the repressor will bind to the operator, has a higher attractive affinity for it, and bind to the operator, prevent transcription of these enzymes, Why make tryptophan if we already have it? And conversely, when tryptophan isn't around, like I haven't eaten in a few days or whatever, I haven't had protein, then the repressor won't bind to the operator. There's no tryptophan around to bind to the repressor. And so it has this low affinity, okay? It has a low affinity for the operator and won't attach and will synthesize, or the E. coli will synthesize tryptophan. Okay. Let's draw this out. Right now, our picture is an active Tripoperon. Okay. And that's how it normally is. Tripoperon. I added that in, italicized. It's referring to the genes. Anyways, okay, so what does this look like? Oh, by the way, I should mention, or I'll mention it in a moment, actually. Okay. So let's draw the regulatory gene. Again, it's far away, and it'll look like this. Let's do it in black. It's okay. The regulatory gene, TRPR, right? So that signifies that it's further, that would be upstream, right? In this molecule, this DNA molecule. So our regulatory gene, TRPR, has its own promoter. RNA polymerase will transcribe it. Just simplify here. So it'll make an mRNA strand, which encodes for this repressor protein, which I'm going to make look like a, let's make it look like a, it's terrible drawing, but kind of this U-shaped thing. Repressor protein. Okay, so we've got transcription, translation. We get this repressor protein from this regulatory gene. And so again, the repressor protein doesn't bind to the operator, it has a low affinity. Unless tryptophan, do in green, I suppose. Let's do a circle here. If there's tryptophan around, it's going to attach to the repressor. And from the slide, when that happens, it induces this shape change in the repressor protein, which increases its affinity, its attraction and likelihood to bind to the operator. So let's draw that. Now check that out. Because tryptophan's around, the repressor has affinity for the operator. It'll bind, and check it out. It physically blocks RNA polymerase from transcribing these genes. These enzymes won't be made. Tryptophan will not be synthesized. Why does that make sense? Oh, well, there's tryptophan around. So that's what I was going to say. That even in, this is why these biosynthesis pathways, a lot of them operate like this. And we're going to talk about why on the next slide. I keep alluding to that, but it's important. Like, why do this? So I'll keep this up, by the way. Tryptophan, if it's not around, we're going to then, you know, the repressor isn't active, and RNA polymerase is going to make these enzymes. We're going to make tryptophan. obviously, right? There's none around. Let's make it. Well, as it begins to accumulate in the cell, it negatively feeds back and shuts down its own pathway because we've made enough. And a lot of pathways operate like this. When the final end product starts to build up, it will negatively feed back on an enzyme in the pathway or a protein involved in regulating it to shut it down. Why? Evolution. Has to do with evolution. Oh, you know what? I think I'm gonna kind of drew this out. So ignore this. Here we go. Important theme. Relate back to evolution. If we're not making tryptophan willy-nilly, then the bacteria, I keep saying we, the bacteria are conserving energy. A lot of these biosynthetic pathways require energy. All of them, they're anabolic, we're building molecules. And resources, these precursor molecules that might be useful in synthesizing DNA in a replicating cell, whatever it is, this is, or other proteins that are needed, etc. This is a good thing. Why? Just because you have a bunch of, I don't know if that works, isn't it? An analogy. I guess it kind of works. Like if you have a bunch of money, it's smarter to buy food than to buy, like you should prioritize food over fancy clothes or a car, right? You don't want to starve to death, but have a really cool car. At least I hope not. But that's the deal. It's always back to this, these bacterial cells that evolved this ability had a better chance of surviving because they were not expending all their energy when they didn't need to, and resources. Okay, one last operon before the end of this lecture, the lac operon. So this isn't a repressible operon, one that's always active, like the Tripoperon. The lac operon, oh, by the way, any questions about Tripoperon, you can shoot me an email. We can set up an office hour, whichever. The lac operon, okay, so the lac operon is not repressible. It's not always active. That is going to be shut down. Rather, it's an inducible operon. Inducible operon. Oh, let's define that first. So an inducible operon is one which is usually off, unlike before, but can be activated when a small molecule interacts with a different regulatory protein. So the lac operon is an inducible operon. So it's usually off But in the presence of this small molecule and another regulatory protein, again, we'll see an interaction. It's not going to shut off the operon that's already on. It's going to turn off the operon that's already off. All righty. So our lac operon. Let's take a look at what's going on here. The lac operon. So again, it's this inducible operon. It's off usually, and then it can be turned on. It can be activated when a small molecule interacts with a different regulatory protein. Okay, so similar to the tryptophan, but instead the molecule isn't shutting it down, and repressing this operon, it's going to be turning it on, and it's already off. Okay, so recall that lactose is a disaccharide consisting of a glucose molecule and a galactose molecule. These are both monosaccharides. The monomer carbohydrates, the monomers of carbohydrates are termed monosaccharides. They're covalently linked together, and lactase is the enzyme that hydrolyzes or catalyzes this chemical reaction, this hydrolysis reaction, right? You needed water and this enzyme to break down lactose and produce glucose and galactose. Okay. So in the absence of lactose, why would we, or the bacteria, why would they make lactase? Again, conserving energy and resources. The enzyme to break down lactose only needs to be made if lactose is present to be broken down. And so researchers have found that when you expose E. coli or any bacteria that have the gene for lactase, they activate this gene to synthesize lactase 1,000-fold in 15 minutes when you expose bacteria to lactose. So we're turning on this gene. It's going to be heavily transcribed and translated to make lactase. Okay. Let's all right, let's begin drawing the lac operon, and then we'll kind of look at the specific mechanism. So I'm actually going to do a similar sort of drawing to similar color scheme and everything to our Tripoperon. Let me go ahead and start with go, we'll do that DNA. And then it's the same sort of situation. We have a lac promoter we have an operator. What I'm drawing right now. And you might find in the text that there's a few genes involved in this operon. But the one we're going to focus on is lactase. You even call it sometimes beta galactosidase. It's the same thing, same enzyme. Just to emphasize the theme that these operons are clusters of genes kind of transcribed together, I'm going to include these other genes, but don't pay attention to them so much. So we have, do it in green. We have 3 notable genes in our lac operon. So 1, and they are called lac Z, lac Y, and lac A. Okay, and so this entire DNA sequence, promoter, operator, the genes, this is our lac operon. Okay. And again, it's usually off, right? So I'm actually going to go ahead and draw in the lac repressor protein. And again, it'll bind at the operator, right? So let's do. There's our lac repressor. And it's physically preventing, again, when RNA polymerase binds at the promoter, won't be able to transcribe downstream. It's always like this. Okay. Great. So now let's talk about what's gonna happen. I think we'll get that. Okay. So LAC-Z is the gene that encodes for the enzyme lactase that breaks down lactose. Again, the other genes, one's involved in transport of lactose into the cell. It's like a membrane protein, but you don't have to know that. And then the other, I think they're still researching. Okay, so again, we're going to have a regulatory gene, LAC-I. Again, it's located outside of the operon, any hundreds to thousands of nucleotides away. Actually Googled in between here when I paused it, I Googled typical bacterial genome size. Ours is like billions in our cells, but 3 billion really, but they range from like 100,000 nucleotide base pairs to like 300 million or 500 million or something. Anyways, fun fact. So anyway, point being is the regulatory gene is located further away from the lac operon, and it's going to code for the repressor protein again. just like before. But it's going to I should not say switch off. Oh, no, it is. It switches off the lac operon, right? Like we just drew. Okay. So before with the Tripoperon, tryptophan bound to the repressor protein, which that activated the repressor protein. But here, the repressor protein's already activated. It's already bound. And so the molecule that interacts with the repressor is going to inactivate the repressor. It's going to allow transcription to occur here by binding to the repressor. And then we'll see that the repressor protein will detach from the operator and transcription can occur. You might think, oh, is lactose that small molecule, inducer molecule that's going to interact? It's actually more specifically allolactose, an isomer of lactose. It's formed when lactose enters the cell. So, not too important, but it acts as this regulatory inducer protein. So when lactose is around, allolactose starts to form through a separate pathway. And it serves the role of binding to the lac repressor, and then it loses its affinity for the operator. And so transcription will occur. All right, so let's draw it all out. Okay. This will be the last thing we talk about today. Questions so far? Again, no one's here, so email. Okay, ask a friend, email, both. All right. So let's start with that regulatory gene, like we did before. And this one is LAC-I. Right? It's not part of the operon. Just to simplify, mRNA is made. It has its own promoter. RNA polymerase will bind there. And we're making this lac repressor protein, right? Which has a high affinity by itself to bind to the operator. And so immediately, this is kind of how it is. It's gonna, right after being made, it just goes and sits on the operator, prevents transcription. Okay. Now, when allolactose is around, because lactose is in the cell, it's going to serve a similar role as what we saw with the tryptophan and bind to the repressor. So I'm going to draw allolactose as a green circle. So when lactose is around, allolactose is formed, and it's going to bind here. Actually, I should draw the arrow the other way. I'm just kind of labeling it. But it's gonna go to the operator here and bind to the lac repressor protein. And what happens is the repressor will lose affinity for the operator and detach. So I'm going to kind of represent that with a red X, because I'd probably use an eraser in real life on the whiteboard, but it leaves, it detaches, it bails. And so this area is freed up for RNA polymerase to then transcribe. The genes in the operator, right, to produce lactase enzyme. And so we'll draw that out as well. To break down the lactose that is now around. And so we can get that glucose and harness it for energy. Okay, so let's write that. So again, we're going to do transcription. The RNA polymerase will be able to do transcription to produce one long mRNA with start and stop codons. I'll even draw in the 5' to 3' ends. So similar as before. And then last, we get translation of these genes. But really the important one here is LAC-Z. So I'll draw 3 blobs to symbolize the proteins. But this one, LAC-Z, encodes for the lactase enzyme, which is what we care about. And that's it. We are regulating gene activity. Keep saying we, but it's the bacteria. But anyways, questions, shoot me an email, phone a friend. If the friend doesn't know, let me know. And that's it. Prokaryotic gene transcription, so regulation. So next time we're going to do us. We're going to do us, plants, fungi, protists, the eukaryotes. How do they do it? It's a little more complex in the sense that this, of course, is complex, but our cells, there's a lot more machinery. There's a lot more going on. And so we have other ways of regulating gene activity. So, all right, until next time.